Thank you. Well, I'd like to thank the organizers for inviting me, especially Juan, and to congratulate you on this 10th anniversary. I know how difficult it is to start something like this de novo, and it's been an amazing success, so I congratulate everyone involved. So what I would like to talk about today is some work that was started in my lab about 10 years ago. And it's about these molecules called the clustered protocadherins. And at the time we started this work, we were very interested in understanding mechanisms of alternative splicing. And a paper came out from Yagi that described a set of cDNAs, which he gave a different name at the time, that had the interesting feature of having the same 3' ends, but having very different 5' ends. So we decided to follow up on that and as a possible model system for studying splicing in mammals. And unfortunately, we could not have chosen a more difficult gene. It's the grand champion of all complex genes, I think, in mammals. So it's shown here, and I'll get into this in a moment. But of course, this gene plays an important function, or genes, and it relates to this problem of cell identity. And so, as you know, individual neurons, in order to have appropriate developmental programs in the brain, need to identify themselves from their surrounding neurons. So they have a unique molecular identity to accomplish that. And it's now known, first shown by Starburst and Drosophila, that this unique identity can be generated through combinatorial expression of individual isoforms on cell surface proteins. And there are two ways that this is done now. One is in alternative splicing, and I'll mention that in Drosophila. And the second is by the mechanism I'll talk to you about today, called stochastic promoter choice. So this is a summary of the DSCAM work, which was mostly done in Larry Starburst's lab over a long period of time. And what makes this molecule so interesting is it's the grand champion in Drosophila. In fact, it's the grand champion, I think, of any gene in terms of its complexity for alternative splicing. And what you can see here is that the genomic DNA that encodes this Drosophila DSCAM has all these different alternative isoforms, 12 of these red exons, 48 of the blue, 33 of the green. And by random splicing can generate a messenger RNA that has its unique red, blue, or green. And that encodes a immunoglobulin domain containing molecule, which you see here, Ig1 through 8. And this turns out to be a homophilic binding domain. There's a transmembrane domain and a intracellular domain. So this gene can encode 19,000 different alternative cell surface splice forms. Different neurons have been shown to express different combinations of these splice forms. And it's shown quite beautifully that self-recognition, that is when two membranes come together that contain a homophilic pair, it triggers repulsion. And so only strong binding between proteins of the same splice form interact, and homophilic binding leads to downstream signaling that triggers repulsion. And that was shown nicely in Drosophila by demonstrating that if you cleave the intracellular domain off, you see homophilic interactions without repulsion. So they went on to look at this at a structural level. And it's summarized here, where you see that if there is a perfect homophilic interaction with 3 matches, you see binding. But if there's a single change, a single isoform change in one of these, you see no binding. And so they've gone as far as determining the structure of the homophilic pair in collaboration with Eisen at UCLA, which is a spectacular structural story as well that is obviously directly pertinent to the biology. Now, in Drosophila, it was possible to look at the mutants in this gene. And what they discovered is if you have 2 wild-type neurons that are in close proximity, they interact with each other, but they don't interact with themselves. Whereas if you have a DSCAM mutant, you can see that it interacts both with another neighboring neuron, but it now forms clumps on itself. So there's a failure of self-recognition, and you see clumping of the dendrites. And if you have a single isoform, you see that it recognizes other neurons as self. So what they were able to show then is that there's this contact-dependent repulsion of homophilic interactions followed by signaling and disengagement. So neurite A and B come together in this way through homophilic interactions. There's signaling receptor ligand displacement there by some mechanism that's not understood. There's a disengagement and repulsion. So the problem with mammals is that it wasn't clear where the source of this diversity arose. And that's because if you look at the genomic organization of the vertebrate DSCAM genes, it's relatively simple. They're capable of generating only a few distinct isoforms. So we believe now, and have recently shown, the best candidate are these clustered protocadherin genes, which, as you'll see, are capable of generating enormous amounts of cell surface diversity. So here are the genes that we identified in 1999, Chong Wu, a postdoc in my lab at that time. And what we discovered when we isolated these genes is that there were 3 clusters that occupy over 1 million base pairs of genomic DNA, and they have a strikingly immunoglobulin-like arrangement where you have these so-called variable first exons, as shown here in their 3 clusters, alpha, beta, and gamma. And you can see that there are all these variable regions. There's a set of constant exons, which I'll describe in a moment. The beta cluster is interesting. It does not have a constant region, but as you'll see, it's highly conserved and has variable first exons. And then the gamma cluster looks very similar to the alpha cluster. So we did a number of experiments trying to determine how diversity might be generated. We did extensive work to see if there were any DNA rearrangements, and there were none. We found remarkably extensive trans-splicing interactions within the cluster, but after 2 years of hard work, discovered that these trans-splicing events, although they occur in cells, that the level of that splicing is so low that it's unlikely to contribute to the generation of diversity. So there's another mechanism. I should say that after we isolated these genes and characterized their genomic organization, we sequenced the mouse in collaboration with Rick Myers several years ago. And you can see from human down to the coelacanth, these genes are highly conserved, both in terms of their sequence and in their genomic organization, and that they have both these variable and constant regions capable of generating enormous cell surface diversity. So we then tried to understand how these genes were regulated, and the first thing we did was to map the 5' ends of the messenger RNAs that are generated in cells. And so I just give this as an example of a case where the promoter upstream from this variable exon called 8 is activated. That generates a protocadherin alpha 8 pre-messenger RNA. And remarkably, even though all of these large intronless exons, and they're very large, contain 5' splice sites at the end, none of these 5' splice sites are recognized. Only the cap proximal 5' splice site is recognized, which splices to the first 3' splice site of the constant region. This generates this protocadherin alpha messenger RNA, which is shown here, which has the variable exon of 8 spliced to the constant exon 1, 2, and 3. And that, of course, corresponds to the protein, which has these 6 extracellular domains. Unlike DSCAM, these are cadherin-like calcium-dependent domains, transmembrane domain, and in a constant, in an intracellular domain. So the question then is, how is the diversity generated? And the Yagi lab in Japan did this set of experiments which revealed the level of complexity that one can generate from this cluster. What they did is to cross two inbred mouse lines, take the F1s, and do individual single-cell analysis. This is way back in 2005, so it was before serious sequencing, and so it was all done by isolating plasmid libraries and sequencing manually. And what you can see is the remarkable diversity that's generated. You see that the two alleles, the maternal allele and the paternal allele, function independently. They are recognized independent of each other. And what you see is that on the maternal allele, for example, you'll see alpha 7 is activated. But then there are these C-type exons, which are different from these, and I'll mention more about those later. And these are expressed biallelically in all cells, whereas in the paternal allele, for example, you see alpha 2 and sometimes alpha 10. So there's anywhere between 1 and very rarely 3 isoforms total that are generated, but they're generated from different chromosomes independently. So it's a very unusual form of monoallelic expression. And so you see that in this neuron, you see alpha 2, 7, and 10, and the biallelically expressed alpha C1 and C2. So you can then see, and we now know that all these clusters behave in this way. They have this random expression. So there are 14 alphas, 22 betas, 22 gammas. And if you assume that the mechanism I showed in the previous slide works universally, which is probably not a good assumption, because we believe there is regulation. I'll show you an example of that. That you can generate on the order of 3 million distinct combinatorial possibilities for individual neurons. So that obviously exceeds by orders of magnitude the diversity generated by the Drosophila DS-CAM system. So we wanted to understand the mechanism by which these isoforms are generated. So we took a classic 1980 molecular biology approach. We identified the promoters, we identified the enhancers, and the binding sites of regulatory proteins that interact with these sequences. So the first thing we could do is by simple sequence alignment, we could see that there was a conserved element upstream from all of these isoforms. And each of them, we could then do 5' mapping of the messenger RNA and show that they map to this site. So there's a transcriptional start site and a conserved element. Now, this is how they look. This is a sequence alignment of all these promoters, and you can see that the same virtually identical sequence is found upstream from all of these sites. Here's a consensus, and the height of the letters here indicate the level of conservation. So this is a very highly conserved sequence, more or less immediately upstream from the 5' start site of each of the isoforms. We did the standard reporter assay using, in this case, a protocadherin alpha promoter fused to a luciferase reporter. Did a series of truncations, and you can see that the CSE is that element that I showed you before. When it's located here, that when it's truncated, you see a dramatic decrease in the level of expression. So this is transfection into mammalian cells in culture. We then carried out a analysis of enhancer sequences, and that was done initially by doing sequence alignments, looking for conserved intergenic sequences. We then did extensive DNase I hypersensitivity assays throughout the cluster, identified a large number of potential sites, and then tested these sites for their enhancer activity, and we found only 2. And they're shown here. And so what you can see is that this is a LAC-Z reporter that is, has in the plasma this hypersensitive site 5 through 1. And it's located here just downstream of the terminal exon. And there's this HS7, which is located here within the intron of the constant region. And when we put that into mice, transgenic mice, you can see that we saw labeling, which is indistinguishable from the pattern of protocadherins, that is, protocadherin alpha is a nervous system specific gene, and I should have mentioned that beta and gamma are also expressed in the nervous system, but they're expressed in other cell types as well. But protocadherin alpha is exclusively expressed in the nervous system. So we then separately deleted these enhancers from mice and looked at the consequences of on expression. And what's shown here is the ratio between the knockout and the wild type, and this is each of the isoforms shown here, and each of these colors represent the levels in various parts of the brain. And what you can see here is that it appears, first of all, that this enhancer is required for maximal levels of activity of all these promoters. Secondly, there seems to be two domains, one from alpha 6 to C1, which is expressed at a high level, and a second is this alpha 1 through 5. And you can see it's highly reproducible, this difference in effect of these enhancer deletions. You'll see here that the C2, one of the biallelically expressed genes, is unaffected by the deletion of that enhancer. If we look at the deletion of HS7, what you can see is a very similar pattern. The effects are not as large as they are with HS5 through 1, but they're significant, and you see again there's this kind of biphasic two-domain interaction, but you see that HS7, in contrast to 5 through 1, is required for the expression of the C2 gene. So we conclude then that this enhancer is required for maximal levels of expression of 1 through 12 in C1, but not alpha C2. There appears to be two expression domains, the alpha 1 through 5 and 6 through 12 and C2. The HS7 enhancer is required for maximal expression of all protocadherins, but like the one above, there are two domains. And what this reveals then is a complex interplay between two enhancers and several promoters. And remember that this is at a single cell level of the expression that I showed you before. So these events have to be occurring, these multiple interactions have to be occurring within a single cell. Now, how do you study that? How do you now study the relationship between protein-DNA interactions and activity when you're looking at the whole brain? And you simply can't, because there are all of these enormous numbers of combinatorial possibilities. So obviously, as a population, you see only 1 in 12 that are active, and 11 out of 12 are silent. So the way we got around this was to look at a very large number of tumor cell lines, and discovered this wonderful cell line called SKNSH, which is very famous, I think, in the ENCODE project. And it turns out to be diploid, and it expresses a set of protocadherins that are indistinguishable from what you would expect for a single neuron. So here's how the protocadherin alphas look. So what you see is that it expresses alpha 4, alpha 8, alpha 12, which you see 4, 8, and 12. That, of course, expresses the ubiquitous isoforms C1 and C2, as you can see here. So this is what you would be facing if you're looking at total brain. So we then did a very detailed analysis of this cell line, both looking in genome-wide ChIP-seq experiments with antibodies that we predicted from in vitro DNA binding studies. And this summarizes an enormous amount of work. What's shown here is that the alpha 4, 8, and 12, C1 and C2, of course I'm showing this as a single chromosome, but some of these are coming from one allele and some from the other. And first of all, you can see that the histone mark for active promoters is only on at the active genes. All these white genes are the ones, the white boxes are the genes that are not expressed. You see none of them have the negative histone marks, the repressing marks. Because of the diversity of expression, we can't really detect Pol2 at these, except for the very highly expressed genes here and at the enhancer. But the most important thing we found was that each of these active promoters binds to CTCF, the insulator binding protein, and cohesin, as you see here. So this is CTCF, and there's a perfect correlation between activity and binding. Here's RAD21, a subunit of cohesin, which you see here. And you'll notice that at each of these sites, at each of the promoters, there are two binding sites. One is directly over the CSE, that conserved element, and the other is within the exon, which I'll show you. So this is a blow-up of that picture. So this is just whole genome ChIP-seq using antibodies against either the H3K4 trimethyl, CTCF, or RAD21. And what you can see is that there are two sites. One, as I said, is located precisely here, and one which was not predicted and is really unique as far as CTCF binding sites. It's within the exon, within the coding region, and you can see it's highly conserved among all the protocadherins, protocadherin alphas. So there are two sites here, and there's enough room between these two sites. It's around 700 base pairs, enough for about 3 nucleosomes that appear to have the transcriptionally active histone modification. So we also looked very carefully at DNA methylation genome-wide. This summarizes a large amount of data, and it basically shows that between the CTCF sites, you see very low levels of methylation in the expressed isoforms. In the silent isoforms, you see high methylation. So there's a direct correlation between promoter activity and DNA methylation. We've shown that in vitro DNA methylation blocks in vitro CTCF binding. And if you treat the cells, the neuroblastoma cells, with azacitidine, which alters the DNA methylation pattern, you see ectopic expression of all isoforms. So there's a very close link between DNA methylation, CTCF binding, promoter activity. You see a similar thing at the enhancer. So here's the ChIP-seq experiments of the HS51 enhancer. And you can see that there are two binding sites of CTCF and RAD21, one located here, one located here. These are roughly the same distance apart of the two binding sites in each of the active promoters. And you can see that in between are these H3K4 trimethyl nucleosomes. So there's, if you will, a symmetry between the binding patterns on the promoter and the enhancer. So, of course, the most reasonable explanation for this is that these enhancers and promoters interact through DNA looping. So we used chromosome conformation capture methods, the 3C method, which is shown here. And this is for using the HS51 sequence as an anchor for the 3C analysis, and this is using the HS7. And you can see that in the control cell lines, K562 and 293, which do not express, you see no looping. But in the cell line SKH-NH, there's a perfect correlation between the active promoters and looping. And you see the same thing is true for HS7, as you can see here. So, indeed, these enhancers interact directly with the active isoforms and only with the active isoforms. If you then do knockdown experiments with a SHGF, CTCF, or RAD21, you can see that in every case, this is now just with the HS51, you see that you lose DNA looping if you knock down CTCF or cohesin. So it's clear that the binding of cohesin and CTCF is critical for the stable looping of these enhancer-promoter interactions. So as I told you, there's an interesting symmetry between the promoter, which has CTCF binding sites of the CSE, and this thing which we called exon CTCF binding site. And there's a symmetry here. And the interesting thing is that the consensus sequences here are in opposite orientations from each other. And so Chung Wu, our collaborator on this part of the work, did an extensive 3C analysis and basically showed that all of these sites interact with each other. And so it's as if There's a at the same time the enhancer can interact with two of these sites and apparently with more than one gene. And so what we're proposing then is that the stability of this complex is facilitated by the so-called double clamp, where there's a Ctcf cohesin bound here, and so this is the enhancer, this is the promoter here, and another Ctcf cohesin here. And then in between is the start point of transcription, the binding site of CBP300, which I didn't show you. There are other transcription factors that bind here, and approximately 3 nucleosomes. So what we're proposing, and obviously this is very hypothetical, is that this stable, and I should say it's very stable. These cell lines have been in culture for 40 years. And we have subclones that were isolated independently at that time, and they express exactly the same set of protocadherins that this does. And so it's as if what's happened is that there's an active transcription hub which brings all of the active isoforms into a region of transcriptional activity. The green parts are meant here to indicate Ctcf cohesin. And you can see that they interact with more than one enhancer. In fact, ChIP-seq experiments involve formaldehyde cross-linking, so if there are extensive interactions, you will, in fact, be isolating the whole complex. And then each of the inactive isoforms are then not in that hub, they're on the outside. So we're now doing experiments to try to address this model. So what about the function? As I said, protocadherin alpha is expressed specifically in the nervous system. Beta and gamma are expressed in the nervous system as well as in other tissues. It's been very difficult to really do careful localization of these proteins. They're actually very low abundance, and it's been extremely difficult to get specific antibodies against these, and almost impossible to get isoform-specific antibodies. But nevertheless, what is published is that there is some localization to synaptic junction, but also all along the cell membranes of axons and dendrites. And the analysis of deletion mutants in mice has been very difficult, due in part, I'm sure, to the complexity of the organization of the genes, but there is some progress. So the first work on this was done in Josh Sain's lab, in which they did a full cluster protocadherin gamma knockout. And what they saw, unfortunately, is that there's a perinatal lethality, lack of coordinated movement, neural cell death and degeneration of spinal cord interneurons, reduced synaptic density, neuromigration, differentiation, and axon outgrowth are all okay. They crossed these protocadherin knockout mice into a Bax homozygous, and you can see it restores, this is a spinal cord section. It restores this, but it did not restore viability, nor did it restore the reduction in synaptic density. So they're kind of stuck. So the next thing they did was to do conditional knockouts, and Julie Lefrave and Josh Sain's lab did a very careful analysis of the retina with this conditional protocadherin gamma knockout. And what you can see is that you see this. So this is the knockout here. You see a reduction in several of the layers of the retina, as indicated here, compared to the Bax homozygous. But you see, there are cells that survive within this layer here, and among these are this internuclear layer, are horizontal and bipolar amacrine cells, and I'll talk about that. But they basically concluded that there's reduced internuclear layer and plexiform layer. They saw loss of interneurons and retinal ganglia cells due to this enhanced apoptosis. So it's been very difficult to analyze these mutations because of the cell death. And Weisheng Shen in my lab was in the course of trying to understand the complexity of the cluster, created a mouse in which only these C genes, so protocadherin gamma C3, C4, and C5, So as I told you, they're expressed biallelically in every cell, and they have different sequences than these. These are the C's in the alpha locus are very similar to the gamma. So what he did was to carry out an extensive analysis of the deletions, comparing it with a full deletion. And I don't have time to show all that data. It's published recently in Neuron. And what he discovered was that the cell death phenotype is entirely due to the deletion of the C genes. And in fact, more recently, a number of papers have come out which are strongly suggesting that protocadherin gamma C3 is a tumor suppressor. It's been associated with a number of cancers. And so, so this part of the locus appears to play a different role. And so you can uncouple the sort of the mechanism of generating diversity from the cell death requirement, the cell viability requirement. Remarkably, the phenotypes of the gamma C and full knockout are indistinguishable at a gross level. Thus, the gamma C is responsible for the cell death phenotype. And so, virtually in every property that we looked at, again, grossly, the gamma C deletion was indistinguishable from the whole cluster. However, now having the ability to, if you will, separate the cell death phenotype from whatever the alternate isoforms are doing, it was then possible to look more carefully, which Julie Lefrave and Josh Sain's lab did with us in collaboration. And she focused on these retinal starburst amacrine cells. And this is a cross-section of how these cells align and tile within the retina. And they have very interesting properties in that they interact with each other in this heteroneural fashion. They don't interact with themselves, so these dendrites here do not interact or cross each other at all in the normal retina. So we then were able, using the combination of these mutations, to look at what's happening, and lo and behold, the gamma genes are required for self-avoidance in exactly the same way as the Drosophila DS-Cam genes are. So here is the heterozygous deletion. Here is the full deletion, and you can see that you see this normal, really beautiful starburst amacrine projection of dendrites, and they're all clumped here. You can see it at a higher magnification. They're crossing each other extensively, and this was extensively analyzed and published recently in Nature. So, but what was interesting is if we now look at what happens, and these are all cell autonomous. They're knocked out only in starburst amacrine cells. And what you can see here is that if you knock out just C345 and you retain all of these, you see a perfectly normal dendritic self-avoidance. Interestingly, and this experiment was done in Drosophila many years ago, where you knock out the whole cluster and then you express only a single isoform, and in this case C3, and you also restore self-avoidance. So anytime you get homophilic interactions, you're able to overcome the self-avoidance. So I'd like to end by just describing some very preliminary work, again by Wei-Xing Shen, in which you see that he's deleted the protocadherin alpha cluster. These mice are perfectly normal. They have a normal lifespan. They display some phenotypic abnormalities of self-mutilation late in life. And if we look at them very carefully with a whole variety of behavioral tests, what was striking is that you see a gross affective function, anxiety and depression, from a whole series of behavioral assays. So these mice clearly have an altered behavior. And what's interesting is that in a paper published by Yagi's lab a few years ago, you can see here that in the dorsal raphe from which the serotonergic neurons project, you see only protocadherin alpha you do not see protocadherin gamma, and we've now done beta. So these serotonergic neurons exclusively express protocadherin alpha, and it's from this site that the serotonergic neurons extend into the rest of the brain. And remarkably, if you look closely at the difference between the wild type, and this is not a deletion, but it's an effective knockout, you can see a very different distribution of serotonergic neurons within the brain, specifically in this CA2 region. And what you see is that in the wild type, there's more or less uniform distribution of these serotonergic neurons, whereas in the knockout, they tend to cluster in one region. And what's interesting is that if you now do a CRG conditional deletion of the protocadherin alpha locus, you see you generate exactly the same phenotype. So you have to express protocadherin alpha within these neurons in order to get normal distribution of serotonergic projections. Now, what's even more interesting is that within the last year, protocadherin alphas have now been implicated in autism. You see here there's a paper that appeared recently. It was this is a SNP analysis where they said protocadherin alpha is a novel susceptibility gene for autism. And there are now 3 studies that came out. This was the first from Mike Wigler's lab, in which they did trio whole exome sequencing, discovered that every individual carries about 200 mutations. And in the autistic children, so this is sequencing the 2 unaffected parents and the child, among the genes that came out were protocadherin alpha. And so obviously we and others are pursuing the possibility that, this locus plays a key role in autism. So the summary is that the protocadherin diversity is generated through random promoter choice and alternative splicing. Promoter choice requires Ctcf and cohesin binding to active promoters and enhancers, which mediate DNA looping and the creation of distinct chromatin domains. Protocadherins are required for self-avoidance and for normal serotonergic projections and have been implicated in autism. And I'd like to thank all the people who did this work. In my lab, Kevin Monahan, the graduate student who's now at UCSF, did all of the work on the ChIP-seq. Wei-Xing Shen has done all the work on creating and analyzing the mouse deletion with the help of a fantastic research assistant, Chiamaka Nwakazi. A lot of the microscopy on the spinal cords that we've looked at was done by Juan Carlos Tapia and Francesco Alvarez. The work on self-avoidance was in collaboration with Josh Sain's lab, mainly Julie Lefrave and a recent graduate student, Dietmar Kostanov. All of the ChIP-seq sequencing was done in Rick Meyer's lab with the help of Kim Newberry, Tim Reddy, and Flo Pauly. And the work on the DNA looping was in collaboration with the Chang Wu lab in Shanghai, Yagu, and Haiyang Wu. Thank you.